So a very good and warm evening from the easternmost corner of India to all the participants. And to some, it's morning, and afternoon to some. So since morning 10 a.m. in India, we have a rigorous session going on from eminent personalities who are in this field, and we have had an interesting and wonderful talks from them on various topics. So my talk will be actually on, yeah, let me share my screen first. Okay. So my talk will be on the taxonomy of Cysorite catfishes. So when I say taxonomy of Cysorite catfish here, my the talk which I am going to present will only limit to classical taxonomic techniques and ideas, not on the integrative as a whole. So in the morning, we have had a talk from Nilesh on integrative taxonomy. So this talk will be mainly a little bit technical, as it will be different from what we have heard in the morning since morning. Then this talk will mainly target those people or ichthyologists who are in the field and who are working in this field of Scissorid catfish diversity and their identification. So, and my talk can be broadly classified into three segments. The first may be on the discussing the techniques of how to identify a Scissorid catfish. And the second segment will be like knowing the various generic characters of some of the Scissorid catfishes which are in Northeast India. I have covered only the genera which are found in Northeast India, but the techniques and how do we identify a Scissorid catfish will be similar all over the world. So it may be followed in that way. Then coming to the third segment of my talk will be on the various hill stream adaptations this small Scissorid catfishes show. And that's why they are also called as the mountain catfishes. So like we'll be coming in that way. So let me start my talk. Yeah, here we can see like two catfishes, Scissorid catfishes. So to a newcomer or to an amateur ichthyologist, it may be very difficult to identify even to the generic level what is this fish. So here comes the importance of taxonomy. Yeah, because if we keep this two, actually the first picture is Cretiochiloglanis peja, and the second is Cretiochiloglanis camengensis. So, but to the normal people and the two new researchers, it will be the same. So how do we identify this? This is the thing which I'm going to talk in the later part of this talk. The correct species identification is the basic starting point of any type of biological study, particularly on the wild populations. For research, it is important that its name applies to only a single species and its species is known by a single name, which was said by Walter Renboth. So which is very true till now. So we need to identify the species correctly. Here comes the importance of taxonomy. Then this is the classification which I followed from Nelson 2006. So this is a general classification of all the fishes. Then here I have marked Actinopterygii. Then from here comes the other Siluriformes. So this other Siluriformes as a whole, the Siluriformes of the Indian subcontinent will be dealt by my colleague Dr. J.A. Johnson from WII, I think on 24th. So I won't be going into that. So I will be dealing only with the family Scisoridae. So under this family Scisoridae, we have two subfamilies, Scissorinae and Glyptosterninae. So this Glyptosterninae are devoid of thoracic adhesive apparatus, and Scisoridae, some members have thoracic adhesive apparatus and some don't have. So coming to the picture of Northeast India, the order Siluriformes, it has got 9 families, and this is according to Kotelett 2013, because Nelson 2006 and Nelson 2016, they included another family Erythistidae, which we think that it is apt to include in Scisoridae as of now, and that we follow Cotlet 2015. And we, like in the morning also, Professor Viswanath talked about the various, how the Himalayan orogeny and the tectonic settings happen and how the drainages are formed and why there is biodiversity in the northeastern region of India. So as we can know, Northeast India is a part of the Eastern Himalaya biodiversity hotspot. And this diversity may be attributed to many factors, which includes the Himalayan orogeny. Then we have the tectonic setting, then the presence of different drainages, which includes the Tinwin, the Barak, Surma, Meghna, Kalagan. Then we have Tista and Karnafuli, which are separate basins. And among the Siluriformes, the family Scisoridae exhibits maximum diversity with around 99 species only in Northeast India. And it is highly ornamental, and some of them are having food value also, so considered as food fishes. And they inhabit fast-flowing rivers and streams. That's why they are also called mountain catfishes, which I have said. And as such, they also show diverse hill stream adaptation morphologically. And even the size ranges from smallest size Erythistis, which measures around like 40 to 42 mmSL, to the largest Bagarius, which can go up to 6 feet. So these are all the members of the family Scisoridae. Then these slide shows us a little insight about where we can find, when we go for field trips, where are the places where we can find the Scisorids. Mainly this Glyptosternans, and with a highly diverse modification we can find in this kind of fast flowing rivers where there are ruffles. So if we go there, then we will get at least Glyptothorax. And if we go higher altitude upstream, then we might find the Glyptosternans too. So these are the various habitats this Scissorid inhabits. Then how to identify a Scissorid catfish? Actually, the Scissorid catfishes, they belong to the family Scisoridae, and the type genus of this family Scisoridae is Scissor rhabdophorus. Then the diagnostic characters of the Scissorid catfishes belonging to family Scisoridae are the following. So any Scissorid catfish belonging to the family Scisoridae will have some of these characters. So the first is body with small unculiferous tubercles. Then the nostrils are very close together and they have a slit-like, and they are separated by the nasal barbel of varying lengths. Then adipose fin may be present, is present always. It may be confluent or not confluent. And it is confluent with caudal in some genera, consisting of a small spine in Scissor. Then the dorsal fin base is short and fin with or without spine, and adhesive apparatus in the thoracic region, it is present or it may be absent. Then 4 pairs of barbels, except in Scissor with adhesive apparatus in the thoracic region present and absent, and 4 pairs of bubbles except in Scissor, which has got only 1 maxillary pair and 5 pairs on the lower jaw. This is the family characteristics of Scissoridae. So a Scissorid catfish will possess some of the combination of the characters given here. Then for identifying a Scissorid catfish, what are the techniques usually we follow, taxonomists follow? Actually, we can go first with morphometry, then meristic counts, then we can go for the osteological studies, then we can, if the specimens are rare, we can go for X-ray radiographs, then even we can for studying the gross morphology, we can go for scanning electron micrographs. Then this part constitutes the classical taxonomic part. And we can integrate always it with molecular, which may be barcoding or molecular phylogenetics, then evolutionary study, then biogeography, and even the river basin thing, which we can add. And it will give a holistic picture, and it will give a clearer picture when we study. But it's not always possible to, combine all this and give a whole picture. Sometimes we need to leave out evolutionary, sometimes we need to leave out biogeography, but as of now, I think it's mandatory to include, it will be best to include the molecular study, that is the barcoding and molecular phylogenetics. So this is the thing, how we can go for the Scissorid fish identification. Then, yeah, for fish identification, first, what do we need? We need the sample, right? We need the material. So we need to go for collection. Yeah, these are some of the pictures when I have gone for collection. This is one in Indo-Burma border, and another sampling site. So this is how we go for sampling. Then, yeah, we go to remote places. And we know that in Northeast India, especially, the roads are not in good shape. Not even there are no roads even sometimes, and there are no vehicles flying. So we have to take this kind of vehicles, which we usually call locally Saktiman. So we have to go here. And if we are doing a night stay near the river banks, we go in a very crude way because it's very interior, and we cannot take like so many things with us, because we have to, after going in this kind of vehicle, we have to trek again for long, like distances. So this is how we go for collection. And yeah. And for identification, what we have to do after collection, we need to identify rate. Then we have to bring it to the lab. Then these are the things we should follow. Like this is the data sheet which we have in Manipur University for counting and measuring. Then the equipments, these are equipments which we require in the lab. Then we have to go for like describing a new species or something, or even identification. We have to browse the literature properly, then osteological studies, then we have to compare it with types and close congeners. Then finally we come to morphometry. And for like measuring a catfish, a Scissorid catfish, we follow Nga and Kotlet 1988. So this is like, for Scissorid catfish, every genus has we have to follow different literatures. We cannot follow one literature for all the Scissorid catfish. So that is the thing. So this is how we measure the Scissorid catfish. Then these are the parameters of morphometric measurement. I hope everyone will be knowing this. Then in case of some like other genera, we need to go for the study of adhesive apparatus. So how we measure the length and the width. For Glyptothorax, we follow Vishwanath and Lintoy-Ngambi. Then even for a new nomenclature which has been made by Hyoki and Kotlet in 2019, so we have to follow this for the nomenclature of the thoracic adhesive apparatus. And this is how we measure the head parts, head lengths. Then for the meristic counts, these are the things we need to keep in mind. So for catfish, we have the dorsal spine. It may be serrated or it may not. So this is the dorsal spine. Before that, we have the simple rays. So this is how we count. Then for the this is the caudal fin. Caudal fin also we have to count. This is the principal ray, which is the longest fin ray. And before that, we have the procurrent rays. Then in some species, we can see like in Cretophiloglanis or even in like the Glyptosternans, they say that we have the spinnet like rays. Actually, this is a picture of Ambliceps, not from a Sisorid, but still I wanted to show the pinnet like rays, how it looks. That's why I just give this picture. Then, yeah, for the pinnet-like rays, this is from Dersal et al. 2001. This is how this pinnet-like rays look in Cretiocyloglanis tawangensis. So this in pinnet-like rays, actually, they are supposed to be in the pelvic and the pectoral in Glyptosternans, but in Cretiocyloglanis, we can find that the pinnet-like rays are also found between the second anal fin rays, simple rays, and the first branch ray. So this is how the pinnet-like rays look in Cretiocyloglanis. Then, yeah, in catfishes, we always have ray fins and adipose fins, and we have the spinous and the branch ray in catfishes. Then for the shapes of when we have to document, we have to write the shapes of the caudal fin. So these rounded, it may be truncated, imaginate. So mainly in catfishes, we have the forked or imaginate or lunate. So these are the things which have to be kept in mind. Then even the gill breakers also serve as a diagnostic features in some of the Sisorid catfishes. So gill breakers, this is the picture from Glyptothorax jeremy. So and this is the redrawn picture. We can see here the gill breakers are born on the epibranch and the ceratobranch. So when we count the gill breakers, it should be 2 plus 1, 2, 3, 4, 5, like that. We have to write. And if a gill breaker is born in between here is this angle, epibranch and ceratobranch, we have to count it with the ceratobranch side. So that is how we count the gill breakers. Then we have to go for branchiostitial ray counts in some species. So this is the branchiostitial ray, and we have to count the numbers which are present. Then, yeah, then the types of mouth, whenever we describe a species, we have to write whether it's a subterminal, inferior, like that, then transverse, whatever. Then the types of teeth are also present. In most of the Scissorid catfishes, we have the villiform teeth, and in some we have the conical teeth also. And they are born mainly on the premaxilla, maxillary teeth. So these are the things we have to keep in mind. Then in catfishes, when we go for sexual dimorphism in Scissorid catfishes, we can see that the urogenital complex in the female, the first picture is of a female. It is mainly a button shape, which is rounded button shape. But in case of males, we can see that this is conical. So the urogenital complex, we can look for the sexual dimorphism. Then in some, we have like skin tuberculations. This is how a skin tuberculations look under the SEM. And this is the pictorial representation taken out from Vishwanath and Lintho and Ambi. This is how the skin tuberculations look. Then when we go for identification of Cisorate catfishes, we have like some characters which are for the family Scisoridae. So osteologically, what happens is the lateral surface of the first dorsal fin pterygiophore and the mesial surface of the distal tip of the neural spine, they have the contacting facets. So this is the first dorsal fin pterygiophore and the neural spine. So yeah, this is how it will contact the fin pterygiophore and the neural spine. Then again, we have here, there is an articular region of lateral ethmoid elongated process as a process and with articular facet for the palatine. So ethmoid process is elongated and the palatine can be, like attached here. Then we have the humerovertebral ligament. Yeah, the humeral process, it is connected to the anterior portion of the vertebral column by a well-defined humerovertebral ligament. These are the features which we see in Scissorid catfish. And here another feature is this The parapophysis of the fifth vertebra is strong and it will be attached to the ventral side of the centrum, directed transversely to the centrum. And this is how we can count the vertebra numbers after staining. And for very rare specimens and types, we can go for radiograph, taking the radiograph. And for studying the gross morphology, like how the teeth looks, then what is this papillation on the upper lip of the glyptothorax, or what is this tuberculations, and what is this thoracic adhesive apparatus made up of. So this kind of gross morphology we can study by taking the SEM. So from the SEM micrographs, we can know easily how they look. So coming to the various genera which is present in Northeast, the first one is Begarius. So the type species of Begarius is Begarius buchanani, and the generic character of Begarius, here we can see the head is covered with keratinized skin. Then the main important thing is the presence of teeth, the 2 or 3 outer rows of numerous close-set conical teeth, and 1 or 2 inner rows of less numerous, widely separated and much larger conical teeth. This is the main feature. And they have this lower jaw markedly with a heterodont dentition, and the pectoral fin with an internally serrated spine, and caudal fin deeply forked lobes. Usually it will be produced as a filaments. And this Begarius, it is the largest Scissorid catfish among the Scisorids. Then coming to the genera Conta. Actually, Conta, Erythistis, and Erythistoides, when we look like just like that, then it looks very similar. People get confused. So what is the difference? We can find out. When I say Conta, when we have Conta will be having, among the 3, Conta will be having adhesive apparatus, thoracic adhesive apparatus, which will be very long and narrow. But Erythistis and Erythistoides, they won't be having a thoracic adhesive apparatus. So both will be different according to their pectoral spine ornamentation, which I will be dealing now. Then in Conta, the pectoral spine, the pectoral spine is with serrations on the anterior margin, which will point towards the tip of the spine, and the lip is papillated. Then head and body are densely tuberculated, and the caudal fin deeply forked with greatly produced lobes. So this is regarding Conta. Coming to Erythistis and Erythistoides, yeah, the difference, both are similar in having thoracic adhesive apparatus absent. But we can see the body structure also in both the genera. Body is short and robust in Erythistis, but it is a little slender and compressed when we go to Erythistoides. Compressed, it will be compressed from the pectoral fin region, not from the head region. Then, when it comes to serration, here I want to elaborate. In Erythistis, anterior margin of the pectoral spine with serrations all pointing towards tip of spine or arranged in a divergent pairs or outwardly directed and not divergent. This we can see in the next slide here. Yeah, in this picture, we can see A and B, they are Erythistis, and C is a spine of pectoral spine of Erythistoides, which I have adapted from Thompson and Page 2006. So we can see the divergent spine here. Then the second one is like it will be pointing towards the arranged and divergent or outwardly directed and not divergent. That is what we can see here. So both the spines are here. I think this question was raised in the morning to Professor Vishwanath. So I want to make it clear to the person who has raised this question. Then another one is Erythistoides. This is the spine. C is the spine of Erythistoides. The anterior margin of the pectoral spine with serrations pointed towards the base of the spine in the proximal half and towards the tip in the distal half here. We can see here, half towards the distal and towards the proximal. So this is how we know easily whether a species is erythistis or erythistoides. Then coming to the three genera, Gagata, Gogangra, Nangra. Actually, when we look at it, people find it little hard to distinguish these three genera. And even Deepina in 1996, he said that they have a derived character, they share a derived characters for Scisorids, and that indicates the formation of a natural group. So that was the thing which Deepina made in 1996. So here we can see here between Gagata and Gogangra, the gill membranes feature and the red dorsal fin feature here. The gill membranes are free but united with isthmus in Gagata, and the red dorsal fin, it is with smooth spine, and they are inserted above the middle of pectoral fin. So it will be like present in the middle of the pectoral fin insertion. Then, in case of Gogangra, we can see that the gill membranes are confluent and also with isthmus, and the red dorsal fin, it is having a smooth spine, but the insertion is behind the pectoral fin base. This is the thing. Then here, Nangra is a little different, so we can find that the body is elongated and terete. Then the palatal teeth is present and premaxilla expanded. So this is the feature of Nangra. But the differences which we have studied is this. So this is the dorsal views of skulls of Gagata, then Nangra, then Gogangra. We can find the characters here, like anterior tip of mesoethmoid. Here we can see that in Gagata, the first picture, it is broad. The tip of the mesoethmoid is here. It is broad and inverted V form. And there is absence of lateral cornea here. This is the lateral cornea which is present in Gogangra, but it is absent in both Nangra and the Gagata. Then here we can see in Nangra also it is pointed and there is absence of lateral cornea, but in Gogangra, the tip is with a V-shaped depression, and we can find two short lateral cornea here. Then anterior fontanel, we can see in Gagata that 1/3 of its length is located in the mesoethmoid region, while 2/3 in the frontal region. But in case of Nangra, we can see that the anterior fontanel, half of its length almost extend almost the entire length of mesoethmoid. And in case of Gogangra, we can see half of its length extend almost the entire length of mesoethmoid. Then coming to the posterior fontanel, which is very different in both the in all the three genera. In posterior fontanel, in case of Gagata, we have half of the fontanel in the frontal region and another half in the supraoccipital. Then we have another is the only 1/4 of the fontanel lies in the frontal region. Then same as Gagata in case of Gogangra. But when we come to lateral fontanel, we can see in case of Gagata, there are 2 pairs of lateral fontanel which are present. And in Nangra, we have only 1 pair of lateral fontanel which is present here. But a lateral fontanel is absent totally from Gogangra. Then the supraoccipital spine, it is elongated and pointed at its tip. Then in case of Nangra, the supraoccipital spine is broad and bifurcated. And in Gogangra, it is similar to Gagata. This we can see in the dorsal skulls of Gagata, Nangra, and Gogangra, the differences. Then coming to Glyptothorax, the type genus of type species of Glyptothorax is Glyptosternum striatus maclellan, and the generic characters are they have got the thoracic adhesive apparatus which are like longitudinal or oblique folds of skin, and it is roughly elliptical, feel like they are on the thoracic region. And there is a long and thin lateral arms of vomer extending the entire length of the articular process of the lateral ethnoid. And there is a presence, of cubital-humeral process, but it is not distinct like in pseudolaguvia. In pseudolaguvia, we can see the cubital-humeral process is very distinct. So these are the various shapes of thoracic adhesive apparatus present in glyptothorax. It may be ovoid, chevron, then leaf shape, rhomboidal. Then this is the adhesive apparatus which reaches the gular region. Sometimes they say reaching the gular region, not reaching the gular. This is how it is reaching the gular region. Then this is the SCM of one of the glyptothorax which I have studied, glyptothorax clavatus, Auravandibula structure. Then in the papillae, we can see the unculiferous papillae with open sensory pores on the upper lip. Then the presence of villiform teeth in the lower and upper jaw in glyptothorax. So this sensory pores also acts as one of the Hillstrom adaptation, because it will be analyzing the chemical nature of the surrounding and also the location of food. This is to compensate the reduced vision in the Sisorid catfishes. So the whole glyptothorax is is a very diverse genus, and we have got a lot number in our teeth. Like it goes up to 31 number as of now. There are 31 valid glyptothorax in Northeast. So we can try to group the glyptothorax. So this is one of the grouping ways which was given by Vishwanath and Linthoengambi 2007. How we can group into three groups of glyptothorax: Glyptothorax manipurensis group, Glyptothorax pectinopterus group, and another one is Glyptothorax cavia group. So this will be on all depending on the adhesive apparatus and the presence of paired fins. So this is how we can group the three glyptothorax, the glyptothorax into three groups, according to Vishwanath and Linthoengambi. But again, when we see other features, we can also group the glyptothorax based on other characters which includes the nature of thoracic adhesive apparatus, which is like depending on whether the medial pit is very deep or shallow, just like the presence of a groove. Or another feature for grouping glyptothorax is whether the premaxilla is having the secondary tooth plate, or is it with the distal piece or not. So these are the two states which we can take. Then the structure of the Weberian lamella. Here we can see like the lateral extension of the Weberian lamella up to the distal tip of the fifth parapophysis. This one, red color. Then another one is portion, or it is shorter. So the state 1 and 2 we can take like this. And another feature which we can take for grouping the glyptothorax is the infraorbital series. So infraorbital bones, some glyptothorax will be having 9 to 10 bones, and some will be with lesser 8 or lesser bones. So this is how we can take the state and group the glyptothorax. Then another parameter is the orbital node, which is present in the frontal bone of the skull. So here the node can be shallow and one can be deep. So in that way we can group. Then coming to the genus Pseudogenus with type species as Glyptosternum sulcatus McClellan. The generic character is body is subcylindrical, then the ventral surface is flat up to the pelvic fin, and the thoracic adhesive apparatus here, it's like different from the glyptothorax. So here it will consist of transverse ridges separated by grooves, and these are respectively called, the ridges are called lamina and the grooves are called sulci, and the caudal fin is emarginate in Pseudogenus. Then pseudolaguvia. Pseudolaguvia, as Professor Vishwanath in the morning also talked about this generic character of pseudolaguvia. The main generic character is the thoracic adhesive apparatus. It is longer than wide and reaching till the end of this pectoral fin. So this is the feature of pseudolagobia. In glyptothorax, it will end here, the length of adhesive apparatus, but it will go until here in some pseudolagobia. Then the presence of conspicuous cubitohumeral process. Here is the cubitohumeral process present here. So this is the main character which we can find in pseudolagobia, and the paired fins in pseudolagobia are not plated and the spine are serrated internally. Then coming to the genus Cesar, actually Cesar is the type genus of Scisoridae. So the type species of Cesar genus is Cisorhabdophorus Hamilton, and it is very distinct from all other Scissorid genera in the absence of mandibular laterosensory canal and the vertebrae with transverse process, and they also have these bony plates on the surface of the body. Then the first principal caudal fin ray is enlarged and elongated as a filament, as we can see in this picture. So, and we can see here the pectoral spine in Cesar, anterior margin of the pectoral spine with serrations. Then genus Cretiocyloglanis with the type species Cretiocyloglanis longipectoralis, Zhou, Lee, and Thompson. So the diagnostic character of the generic diagnostic character of Cretiocyloglanis is the presence of interrupted postlabial fold. Here it is interrupted. Then broad isthmus and gill openings. It will the gill openings will not extend to the venter ventrally. It will not be there. Then the upper jaw patch tooth jaw, the jaw tooth is patch is joint and not produced posteriorly on the sides. So these are the generic characters of Cretiocyloglanis. Then I think Professor Vishwanath also explained about Meyersglanis and exostoma in the morning, their the structures of their oromandibular structures. Here we can see this is a Meyersglanis. So the postlabial group is continuous. Then the gill openings are not extending to venter, and they have a distally flattened teeth in both jaws. Here we can see the shape of the teeth. This was taken when I was studying Meyersglanis. So the teeth is oar-shaped and large and movable with longitudinally curved inner face. Then the tooth patches are joined in the upper jaw here. The upper jaw, the tooth patches are joined. Then we have the homodont dentition in both the jaws. And this is the laminar adhesive pad on the ventral surface of the paired fins. Here we can see unculi. It is differently arranged. Here we have the globular unculi at the anterior region, then stout and densely packed in the middle. So this is how the arrangement happens in the ventral surface of the paired fins. This is actually the plique, which is present on the ventral surface of the paired fins. Then exostoma, the type species Exostoma Bertmori, Blit. Then the characters here. So there will be always a question on whether it's a Meyersglanis or it's an exostoma, because it will be difficult to identify at an instant whether it's a Meyersglanis or an exostoma. So we have to go and see whether there is a presence of interrupted or uninterrupted postlabial groove. Then we have to see the jaw and the tooth patches and what kind of teeth are present. So these are the things which we have to take in mind while identifying Meyersglanis and exostoma. Then this is genus Oreoglanis, type species Oreoglanis siamensis Smith. So here also we can see the presence of continuous labial fold, post-labial group. Then there is homodont dentition in the upper jaw. So it will have a pointed dentition in the upper jaw, but in the lower jaw, it will have two types of teeth, truncate and spatulate. So it will be a heterodont dentition in the lower jaw. Then, yeah, the slide shows how the current is there. I mean, how fast the currents are in the hill streams. But in this kind of environment, the Sisorid catfishes are surviving. So they have these special features for adapting to this kind of life. So what are those? We have already seen that this is the external form, how they look like. They are leaf like. The ventral is totally flattened so that they can attach to the substratum, so that they are not carried away by the water currents. Then they are relatively small size. This is advantage for hiding underneath the stones and the pebbles. Then we can see the shapes of caudal fin. The lower lobe is longer so that there is increased surface area of contact. Then the caudal peduncle is slender so that there can be rapid movement against the water current. Then the eyes are very much reduced, which is a hill adaptation to hill stream, which is because the hill streams have shallow water. And moreover, to compensate this, as I have mentioned earlier in one of my slides, that they have this the maxillary the barbels. They have got this sensory pores, so they will sense the chemical nature of the water in the surrounding and even the predators. So that is how they compensate. Then the eyes are always present on the dorsal surface, not on the ventral surface, because the ventral surface will be used for adhesion to the substratum. The gill openings and branchiocedial rays, there will be more numbers, and gill openings will be broad. Then we have this one, the Auravandibula structures, mouth situated ventrally, and the jaws are very strong, and the barbels greatly reduced, short and stumpy. And in glyptosternum, mouth is spread in broad sucker type so that they can attach strongly. Then these are also one of the apparatus which they use for adhesion, thoracic adhesive apparatus. Then the paired fins. The paired fins has got pliques underneath. So these are also for adhesion and for locomotion in those like fast flowing waters. Then, this is the SEM studies of different unculi which are present in the thoracic adhesive apparatus of 5 species of glyptothorax. We can see how they differ under the SEM micrographs. So this is in Pseudochinus, and the unculi in Pseudolaguvia, we can see here, they are very long and curved and hook-like. So this is how they are used as an adhesive apparatus. Then these are the features which we can see in Exostoma and Mears glanus. Then this is the Auravandibula structures which are found, we can see in Pseudochinus, Mears glanus, and Exostoma. Taste buds are also present on the barbels. Then, which are present in the upper lip. This is a feature which is present in unculiferous papillae with open sensory pores which are present on the upper lip of glyptothorax. And this is the taste buds on the barbels which we find in Pseudochinus. So they are like mainly for adaptation to hill stream mode of life. That's all. Thank you.